Hey everybody, welcome back. This week I'm going to be introducing lab number 8, which is chapter 9 in the lab manual, where we're going to be looking at late Paleozoic life. So last week we looked at early Paleozoic life. We introduced phylum such as cnidarians and mollusks and echinoderms and arthropods. We're going to look at echinoderms today. But we're going to be revisiting some of those same phyla from last time and just kind of picking up where we left off as we move through time. So it'll be the same kind of thing as previously, where we'll do this short video, you do the pre-lab, do the lab exercise, and we'll talk a little bit about the upcoming quiz at the end. So just like last time, here's kind of a good snapshot of what the world was like during the last half of the Paleozoic. So the late Paleozoic includes the Devonian, the Carboniferous, which is the Mississippian and Pennsylvanian periods, and then the Permian. So the late Carboniferous would be the Pennsylvanian period. And as you can see at this point, Pangea has pretty much started to form. North America is still rotated a bit and straddling the equator, but we've now converged with Gondwana, and we're starting to form. This is like the last death throes of not having a supercontinent. So everything's starting to come together. As this happens, global sea levels start to fall. We're uplifting a huge mountain range. We're seeing an increased amount of aridity and dry conditions around the world, a lot more deserts, and a lot more sediment coming off of those new mountains, filling in some of these shallow water basins. And this is putting a huge impact on life. So life is having to move not only from water to land, but the ones that stay in the water have to come up with some new strategies because the water is really murky with lots of clay and silt. And if you're a filter feeder, like most of these organisms are, that produces some challenges. So last time when we looked at our slough sequences, we focused on the Sauk and Tippecanoe sequences during the early Paleozoic when sea levels were generally much higher. However, in the last half of the Paleozoic, Devonian through Permian, we're looking at the Kaskaskia and Absaroka sequences. And at this point, well, the Absaroka is kind of weird. It looks like sea level's high. It's a very thick sequence, but that also includes desert deposits, so the complete opposite of high sea levels. So we have a very thick Absaroka sequence, but not much of that is actually marine deposit. So just like last time, let's start looking through the different types of animals, and in this case, plants too, that we're going to be looking at. And we're going to start again with kingdom Animalia, and this time phylum Echinodermata. I've talked a little bit about echinoderms in lecture, but since we'll be looking at them again in lab, I figure we should cover them again. Echinoderms are really weird, frankly. They don't have blood. They don't really have organs. They pretty much move through hydraulic power from seawater. They pull water into their bodies. They have a whole bunch of tubes running through their bodies, and they squeeze those tubes, and that pumps water from one place to another, and that causes movement. So they all act like big hydraulic organisms, except most of them are actually pretty small. So phylum Echinodermata, this also includes your sea stars, your brittle stars, sea cucumbers, sea urchins, sand dollars, and then these really weird things, the blastoids and the crinoids. These are our two main classes of Paleozoic echinoderms we're going to look at. Blastoids are what you see on the far left. That's known as a Pentromites or a sea bud. And those 5 regions that you see on the top view, we call those ambulacral areas, and they have pentameral symmetry, 5-fold symmetry, just like a sea star, just like a sand dollar, just like a sea urchin. These were suspension feeders. Most of our Paleozoic echinoderms are suspension feeders. They'll sit there and just with these little fibers or what we call pinnules sticking out, and they wait for something to touch them. And when it does, they will push that towards the mouth. So the 5 little dots you see in the center of that Pentromites, that blastoid, are actually the mouth of this thing. And then we have crinoids, which are much more diverse. Crinoids look like plants, but they're not. They're animals. They have a stalk and a crown, which looks like a stem and leaves. They have a holdfast, which looks like roots, but all of these are flexible and can move around. So crinoids would branch out their arms and extend their pinnules and just kind of lazily sway in the water. And when a small piece of organic material would fall through the water column and land on it, those pinnules, which are individual calcite crystals, start moving that food kind of hand over fist down the arm to the arboreal cup where it's eaten. Now, on the far right of the screen, what you're looking at are calyxes, or the arboreal cups of crinoids. So the other word for arboreal cup is a calyx. And again, notice the 5-fold symmetry. That's a common thing with echinoderms. They all come in these 5-point symmetry shapes. Now, when most echinoderms die, they fall apart because each piece is an individually grown calcite crystal. Again, they're really strange. They have crystalline bodies powered by hydraulics, and they look like plants, but they're animals. And when they die in that soft tissue, the very thin coating of soft tissue breaks down, all the pieces tend to scatter. So we have lots of limestones made entirely of echinoderm pieces, especially crinoids. Not all of our animals from the late Paleozoic are large. We're actually going to be looking at the largest form of the smallest kind of animals today as well. These are within phylum Protozoa, so they're protists. These are Fusulinid Foraminifera, or Fusulinid forams, or just forams for short. We still have plenty of forams alive today. Most of them are about the size of a grain of sand. They're tiny little single-celled organisms. They're amoebas that grow a calcium carbonate shell. What makes Fusulinids interesting is that they're the size of a grain of rice, which is pretty small, but for a single-celled animal, that's enormous. That's like you carrying your house on your back. They're really, really big for a single-celled organism. Now, they're a benthic foraminifera, which means they live at the bottom of the seafloor. These things are too big to float around freely like most foraminifera do. And if you take the Fusulinids, these little rice-shaped fossils, and you cut them in half, you can see the internal coiled anatomy of these things, where the amoeba was living kind of inside and around this shell hard part. So these are used for identifying sea surface temperatures, or at least bottom water temperatures, for the late Paleozoic. So paleontologists will find these little tiny foraminifera, and they're made of calcium carbonate, so we'll analyze the calcium carbonate, and the chemistry of that can tell us something about the water that was around at the time this organism was forming that shell. So modern-day foraminifera and these gigantic Fusulinids are really, really important for understanding ancient ecosystems and also ancient climates. Last week, we looked at Superphylum Lophophorata, and we did look at brachiopods. So we focused more on the inarticulates like Lingula, but the articulate brachiopods are very, very abundant in the late Paleozoic. Most of them go extinct at the very end of the Paleozoic. We only have a few of these left alive today. The two types of articulate brachiopods we're going to look at today are the sporiferids and the productids. So we have a shallow water environment because it's the late Paleozoic and sea levels are falling. And we have huge mountain ranges shedding sediments off, and that sediment's going into these shallow water environments. Now, brachiopods are filter feeders, which means most of what they're filtering is mud, and that's not going to work very well for these guys. So they have some kind of cool adaptations for how to handle this. The sporiferids evolve a very long hinge. In fact, you look at the bottom specimen here of the sporiferid, and it's kind of D-shaped, where it's flat on the bottom and then curves on top. That means that lophophore, its filter feeding structure inside, has more space to coil outward. And we call the structure at which that lophophore holds onto a brachydium. And if you look at the image to the right, you can see a mineralized, fossilized, coiled brachydium inside of a brachiopod shell. So sporiferid brachiopods evolved larger filter feeding apparati, larger lophophores, so they can filter out more mud and still get some food. Now, the productive brachiopods have a new adaptation. If you recall from last week, brachiopods anchor themselves in the mud with a fleshy stalk called a pedicle. They stick that in the mud, and then they sit there and they open their mouths and filter feed. The productids find that with so much mud sticking around, they can't anchor themselves in. It's like trying to put a strong stake into sand or something. It's just not going to keep things upright. So they do away with the pedicle altogether. They just get rid of it, and they evolve long spines coming off of their shells so they can anchor in with a whole bunch of stakes rather than just a single fleshy appendage. And this works really, really well. So they can't really get up and move around very easily, but they can anchor themselves into place and stay there during that shallow water wave action isn't going to knock them out of place. Now, most of the times when we looked at productive brachiopods, the spines break off because they're like calcium carbonate needles. They're very, very thin. But they probably weren't used for defense because they're so delicate. But in fact, they do look like they grew in response to allowing this thing to anchor or grip onto a substrate so they could do their filter feeding. And just like we just looked at brachiopods and how they've changed from early to late Paleozoic, the bryozoans go crazy. We have a new group of late Paleozoic bryozoans called the phenestellids. Now, the word fenestra or phenestella is Latin for window or opening or vestibule, any kind of opening. The phennostellate bryozoans have these big lacy nets, except when these were first discovered, they weren't always associated together because there's two main parts of the phennostellate brachiopods, especially the genus Archimedes. You have a screw-like fossil, and it's seriously the size and shape of like a wood screw, and then these lacy nets. Turns out these are all part of the same colony of bryozoan. That the screw axis was the center, and these lacy nets would wrap around and stick out like fronds, as you can see in the reconstruction. Now, what's the function of this? Again, muddy sediment with a moderate amount of energy, keeping that mud and silt suspended. This allows a pre-filter for our filter-feeding bryozoans. So as water full of silt and muck would wash through these fronds, inside those frond baskets, the velocity of the water is going to be slower. It's going to slow that velocity down. That means the sediment's going to start to fall out of suspension. Meanwhile, the bryozoans, which actually are living on the crossbars of those nets, they're actually living in those individual crossbars, can filter feed out any organic material. So these are some of the adaptations these organisms take to overcome a changing environment from shallow water, or from deep water to shallow water, from clear water to more murky water. It's also during this time that a lot of our insects and vertebrates are moving from the oceans onto land, which is something that we've talked about in lecture. And one of the other things that does this are plants, which you can see behind me, some random images of late Paleozoic plant fossils. A lot of the plants that we see in the late Paleozoic are very, very strange. If you could go back to the Pennsylvanian and look at some of their forests, they look totally alien. We have trees that have scales on them, similar to what you'd see on a reptile, except they're made out of bark or very thick, kind of a cornified material. And one of the purposes of bark or a waxy cuticle over a plant is to help keep water inside the plant. So our scale tree, which we call Lepidodendron, were pretty large. And then we have smaller trees with weird little branching leaflets known as Calymites, and some of these leaves, which we call Glossopteris, which actually look like a tongue or a feather. So it looks like ferns on trees, which is kind of strange by today's standards. But this is when we start seeing true rainforests and seeds. Today, most of these rainforests are preserved in the coal that we mine in Pennsylvania, West Virginia, Southern Illinois, and parts of Iowa. That's why we call them coal forests. So when you hear about the coal mining going on in certain parts of the country, that's Pennsylvanian aged. That's where we get the name Pennsylvania. It's where we get a lot of these types of fossils. This is also the time when we had gigantic insects, big dragonflies and millipedes the size of surfboards. And the reason for that is all of these new forests are producing lots of oxygen. And the arthropods breathe through the segments in their exoskeleton. So the more oxygen, the bigger they can become to accommodate that. So we have giant arthropods, we have giant forests, and this is also when we're starting to see giant amphibians coming out of the water. So arthropods and insects actually dominated the land first, and then as vertebrates got larger, we kind of saw this little bit of an arms race and things balancing out. So as you begin Lab 8 on late Paleozoic life, a couple of things to note. Again, make sure you're reading through the lab manual just like last time, and please do the pre-lab assignment on Canvas before you get started on the actual lab itself. Just some good critical thinking questions. Utilize your lab manual for Appendix B on page 195 and the study guide on page 292 as we prepare for the quiz next week. So again, I originally planned on doing a quiz for this class where we would pass fossils around. Obviously, we can't do that right now. So it'll involve a lot of images of fossils with some drop downs and answering questions about those. So some things I want you to focus on is the classification and some of the features and life modes that we've talked about and that you have read about for these groups of organisms. However, you do not need to identify them to the genus level. I won't be asking you what genus of trilobite is this or what type of plant is this. Instead, you'll be asking more about the features, the anatomy, and when they lived, okay? So classification, important anatomical features, and when these individual organisms lived. And all for that, I'm looking for early or late Paleozoic. We either looked at it last week or you're looking at it this week. It's that simple. So here's an example. Perhaps you on the quiz, you would see an image of a trilobite such as this, okay? Name this class of arthropod. The answer would be class Trilobita. What feature is highlighted? Okay, what feature is that? Well, that's the cephalon. And when did this animal live? Early Paleozoic. So the questions are going to be things just like that. So if you start looking through those 3D PDFs, remember those are still available, and you look over the labs a few times, you can go over the lab repeatedly, and using your lab manual, using that study guide, you should be good to go. So until then, good luck on both the lab and on the quiz, and we'll talk to you all later. Hope everybody's doing well.